Hello, everyone, and welcome to our one-on-one discussions with the scientists within the MAMI project. My name is Dalton Dietrich, and I'm the scientific director of the project. And today, we have the pleasure of speaking with Dr. Damien Pearse. Damien is a full professor in the Department of Neurological Surgery, an extremely important component of our MAMI project family and research activities. So, Damien, how are you doing today?
Very good, thank you. Thank you very much for this opportunity to talk about my research.
Fantastic. So, Damien, cell transplantation. You've been doing a lot of transplantation for all your research career. So tell us why cell transplantation is so important when we think about treating spinal cord injury.
So often after spinal cord injury, the damage to the spinal cord and the loss of cells results in the formation of a cyst within the spinal cord. And this cyst is generally filled with fluid and immune cells, and because of that, the axons find this cyst as a physical impediment to grow across the injury site. So in order to allow these axons to grow across the cyst, we need to fill it. And cell transplantation is one way that we can replace those cells that have been lost after spinal cord injury and provide a substrate upon which the axons can regrow and reconnect to the caudal cord.
So you're transplanting the cells in a cyst or adjacent to the cyst. How clinically relevant are our models to mimic what we're trying to treat in people with spinal cord injury?
Yeah, so these cysts are quite large in human spinal cord injury, and they're quite variable. So sometimes individuals who have spinal cord injury may develop a cyst that could be several centimeters in length that could go over several segments of the spinal cord. And sometimes there may not be a cyst. Sometimes there may be demyelination and damage at the region of the spinal cord, but not the development of a cyst. However, for our experimental models, we can't have a model that's as variable as the human condition. So generally, we use a very reproducible model in which we provide an impact to the spinal cord, generally using a weight drop or a force, and this results in the formation of a cyst. And we use very reproducible injuries. We monitor the parameters so that every injury is the same. And this is important so that we can compare animals and look at the treatment effects of a therapy. And so in our laboratory, we've developed a multitude of different models to look at spinal cord injury that replicates the human condition. So if we look at the human condition, the majority of individuals are males. They have injury to the cervical spinal cord. And generally, they're at least 1 year out from when they receive spinal cord injury, so chronic injuries. And so in our laboratory, in collaboration with yourself, we developed one of the first cervical contusion injury models. And this is a bilateral injury, so it damages the spinal cord in a way that's very similar to the human pathology. And it forms a cyst that we can then use to transplant cells. And we've also started most of our work now in chronic injuries, and this is where we transplant the cells within 2 to 6 months post-injury. And we know that in chronic injuries, there's a reduction in the ability of axons to regrow because there's a dampening of the regeneration-associated genes that are important for stimulating axonal growth, as well as the development of a very robust scar, which is a physical and chemical impediment to axon regeneration. It contains many inhibitory molecules, such as chondroitin sulfate proteoglycans, as well as reactive glia. And so the chronic injury model, if we find therapies that can improve regeneration, improve outcomes, they'll be amenable to the majority of people who suffer from clinical spinal cord injury.
Thank you very much. So you can do cervical injury, thoracic, you could do lumbar injury, you could do different injury severities, and then you're trying to understand if your treatments affect function. What type of functional outcome measures are important that we can carry out in animal studies and translate to people?
Yeah, so the majority of people who are injured with spinal cord injury, they either are paraplegic or quadriplegic. Up until recently, most of our studies focused on thoracic injury, in which we were looking at paraplegic animals or animals that had loss of function of their hind limbs. Unlike humans, rats have very rudimentary walking functions, and so it's very difficult sometimes to look at functional outcomes of the hind limbs. However, with the development of cervical injury models, we can now look at the forearms and the forepaws. And these are much more amenable to very fine tasks, which is similar to what a quadriplegic individual would be doing, such as manipulating a cup or manipulating an object. So we can use very skilled motor functions in our animals and look for fine improvements in outcome with the therapy over time. So these cervical models are much more similar to the clinical condition as far as functional outcomes.
So we've talked about cellular therapies, but there's a lot of different types of cells that we and other laboratories throughout the country are utilizing in their transplantation strategies. So can you tell us about some of the cell types that you're studying today?
Yeah, so initially my work started off in the laboratory with using swan cells. Swan cells are a glial cell that's very important for peripheral nerve regeneration. It's one of the cell types or a critical component of peripheral nerve regeneration that is successful in restoration of function. And these are nerves that are found in your legs and in your arms and your fingers. And so work by Mary and Dick Bunge showed that they could develop swan cell culture methods and use these cells for transplantation into the central nervous system. And so when my laboratory started, I continued this work, and I've been working with swan cells now for almost 20 years and about 15 years within my own laboratory. And initially those studies with swan cells looked at the protective effect of swan cells, how the swan cells could improve outcome in a chronic injury model. We showed that when we transplanted swan cells into a chronic injury 2 months after injury to the thoracic spinal cord, that we could get supraspinal axon growth into those transplants, and that we saw an improvement in function. And then following the generation of that data, we were interested in moving swan cells towards clinical trials. And my laboratory participated for about 5 years in developing investigational new drug application data that we could submit to the FDA to show that swan cells transplantation was feasible and that it was safe in chronic and acute spinal cord injury. And then we were able to use this data to initiate 2 phase 1 clinical trials in subacute and chronic human spinal cord injury, of which the first subacute trial is completed and the second chronic trial is still ongoing. But so far the data looks like they are safe in those paradigms. And so in addition to swan cells, some of my work has looked at comparing swan cells to olfactory and sheathing cells. And these are a glial cell that are found in the olfactory bulb near your nose. And they're very important for guiding axons in the development of the olfactory bulb. And we compared these cells to swan cells. They exhibit many of similar characteristics as supporting axon growth. However, we found that their survival was more limited than swan cells, and that they didn't promote as much axonal growth as swan cells. Recently, we've started to work with neural stem cells. Neural stem cells have been shown to, when transplanted into the spinal cord, generally have a fairly low survival rate unless they're differentiated towards an oligodendrocyte lineage. And these neural stem cells, because they have a low survival rate in the spinal cord, they've been combined with extracellular matrix, neurotrophins, and cell death inhibitors so that they can survive and persist. And so these results with neural stem cells from other groups have shown that they can differentiate into neurons and form a relay across the spinal cord in which they can allow for communication between the brain and the caudal spinal cord. So one of the studies that we're looking at the moment is to use co-implantation of 2 different cell types that have functions that could be additive or synergistic for repair. So if we transplant swan cells into the spinal cord, these cells within the lesion site have very good survival, and they modify the lesion site to one that is pro-reparative. And if we can use that graft and then transplant neural stem cells into this environment, maybe they'll be able to survive better and then be able to differentiate and form a relay, and the swan cells can myelinate those axons that are growing in the relay to improve axonal conduction. And so far from some of the chronic studies that we're doing, we've seen very interesting results. We've been able to see that both the swan cells and the neural stem cells survive at very good levels, very robust filling of the lesion cavity. So we're excited to continue those studies and look at whether there's any functional improvement or improvement in regeneration and repair.
Very, very interesting. Thank you. And now the technology is available to actually maybe manipulate the cells and maybe make them release factors more or survive better or migrate better. So can you tell us a little bit how you're manipulating the cells to have a better result?
Yeah, so one of the issues with a lot of cells that are transplanted is that they get restricted at the lesion site. And if the cells are restricted at the lesion site, they cannot migrate out where they can reach axons that are retracted after the injury. This is very important for descending tracts like the cortical spinal tract, which when severed, it retracts from the injury site. So if the cells can't migrate out of the lesion, they can't reach those axons and guide them across the lesion. Also, if they can't migrate, they can't guide those axons then from the lesion into the caudal cord towards their targets. And they can't reach areas that may have distal demyelination to mediate remyelination repair. And this is a property that's associated with Schwann cells, because Schwann cells, they're a peripheral nerve element. They cannot migrate into the central nervous system normally because they're restricted at the dorsal root entry zone. And they're restricted by molecules that astrocytes secrete, such as chondroitin sulfate proteoglycans, ephrins, and they're also restricted by the physical impediment of the cells themselves, the astrocytes. And so when we transplant spinal cords, transplant Schwann cells into the injured spinal cord, they become restricted by the scar and they don't migrate out. And so we've been looking at a molecule called polysialic acid. And polysialic acid is a molecule that's expressed on the surface of cells. It's expressed very highly during development. And it's involved in when these initial neural cells migrate out from the neural tube to form different regions of the brain. And in the adult, it's expressed in areas that have very high neuroplasticity, such as the hippocampus. And so in our original studies, we looked at using a gene therapy approach to express this molecule on the surface of Schwann cells. And what we found is when we expressed polysialic acid on the surface of Schwann cells, that they then were able to migrate out from the lesion site and associate with supraspinal axons, such as the cortical spinal tract axons, and guide those axons not only into the lesion, but from the lesion into the caudal cord and improve functional recovery. And so we're now working with collaborators in Canada and New York and California to develop a biological method using an enzyme that can allow for a very transient expression of polysialic acid on the surface of cells so that they can escape the lesion environment and be able to interact with axons and guide them across the lesion. So we're very excited about using this enzyme in chronic spinal cord injury and testing it against the gene therapeutic approach that we've used previously.
Very exciting, very exciting. I'm always interested in hearing you talk about some of the new innovations that you're utilizing that are building on the knowledge that we have in hopes of actually doing a better job of repairing the nervous system. Now, we talked today mostly about cell therapies. What other promising therapeutic interventions is your lab working on today?
Yeah, so in addition to looking at cell transplantation, we're looking at a number of different therapeutic avenues. One avenue that is very similar to cell transplantation is using small membrane nanoparticles from cells called exosomes. And so exosomes, these are membrane particles of a nanometer size, and they're produced by cells, and they're involved in communication over distances that are larger than what would normally occur with secreted proteins. And so exosomes, they generally contain receptor signaling molecules on the membrane, as well as genetic material inside the exosomes. And this genetic material is normally microRNAs, and when it binds to a target cell, these microRNAs are released into that cell, and they reprogram the cell towards a particular function. So for example, a neuron that's under stress, if these exosomes bind to that neuron from, say, a glial cell, they may have microRNA that contain anti-apoptotic factors or pro-survival factors, or they may contain microRNA that can stimulate regeneration and regrowth. And so we're looking at exosomes and seeing how best to deliver these small membrane vesicles, how to develop their extraction from GMP cultured cells that we use clinically, and how we can determine whether these exosomes may have reparative efficacy that is as good as the cells from which they're derived. And the benefit of exosomes is, unlike a complex surgery where we may need to open the back of an individual and expose the spinal cord and transplant the cells into the injury site, exosomes may be delivered systemically. They may be more amenable to repetitive injections or administrations. And so it may be an interesting avenue to explore beyond cell transplantation. We're also interested in trying to make the spinal cord more receptive to cell transplants. And so previous studies had shown that many of the growth factor receptors that are expressed on axons are downregulated or absent in chronic injury. And so if we want to use growth factors in addition to cells to promote regeneration, the receptors may not be there to signal to the axons to grow. And so we're looking at ways that we can have these axons re-express these growth factor receptors so that they're more receptive to the cell transplants that we put in with growth factors. And so this may be a more robust regenerative or reparative strategy that we can use a combination of approaches.
Okay, this has been very stimulating. It's obvious that your laboratory and your colleagues are doing a lot of exciting work that's going to hopefully make a big difference in people's lives living with spinal cord injury. Damien, thank you for joining us today. Congratulations on all you and your colleagues are accomplishing, and we'll have you back to hear more about the great progress you're making in the laboratory. Thank you.
Thank you very much. Have a good day.